we have about 300 folks registered, 160 in person and the rest online from about 80 institutions from around the world. So the Center of Engineering Mechanobiology is a science and technology center of the National Science Foundation. So these are some of the biggest investments that the National Science Foundation makes, typically at the interface of different disciplines. So in our case, it is life sciences and physical sciences. So our center was first funded in 2016. But like everything that government does, we didn't get our funding till 2017. So we are kind of pushed back by 1 year roughly. And about 2 years ago, we were renewed for a second 5-year period. And so we are a partnership of 10 institutions led by Penn. We have partners throughout the country. Now, our center was established based on the idea that the specifics as well as the general principles by which plant and animal system respond to force can inform each other, and the fact that these things can lead to technologies. So that's the basis on which we founded our center. And our vision is to develop a multi-scale approach. When I say multi-scale, it means going from molecules to cells to organisms. To study forces in living systems. When I say living, it means plant and animals, and to train a new generation of scientific leaders in this discipline. Now, our center is as far as I know, is the only center in the world where we are using the mechanical response of cells to address grand challenges in human health, as well as for growing and protecting crops. So in our first phase of the center, the idea was to understand how forces were generated in the cells and the microenvironment, and how this went all the way up to the nucleus, and how that sort of led to changes in gene expression patterns. So our efforts were focused on 3 fundamental questions that cut across plant and animal systems. How do cells sense force? How do cells adapt to force? And how do cells remember force? So in our second phase, which started about 1.5 years ago, the focus has been on what we call mechanointelligence. This was a term very intelligently coined by Trina Lindsay in tomorrow. So the whole idea of mechanointelligence is basically trying to figure out how cells are able to sense, respond to, and adapt its own properties as well as the properties of its mechanical environment for enhanced fitness and function. So broadly speaking, intelligence really involves first sensing and adaptation, which typically takes place at the adhesions, at the cytoskeleton, and perhaps at the nucleus. And then learning and memory, so where the nucleus plays a very key role. Now, our efforts in the second phase is focused on focus around 3 what we call interdisciplinary research teams. And the first team will, as you will see, will address how the sort of the recursive dialogue between the cell and the microenvironment. The second term is on focused on the nucleus, where we are looking at new ways to implant and exploit memory at the level of chromatin and the nucleus. And finally, 4 is using all these things to develop systems where we can emulate tissue function in 3+1 dimensions. Now, I'm not going to go much about our work because it's going to be covered in beautiful talks by Malike Lakaramiali on the nucleus, Becky Wells on the recursive dialogue, and Trina Aridze on tissue systems. So just to give a little bit of preview of their work, I just have one slide. So cells may sense their environment, which may be stiff or elastic, or it could be soft and viscous. And based on this, the cells will adapt their cytoskeletal machinery or chromatin in the nucleus on short time scales, typically associated with mechanical chemical signal, maybe minutes, maybe hours. And so in the first IRT, you will see, you hear more about the recursive dialogue, in particular, focusing on properties beyond elasticity, viscosity, plasticity, disappearing mechanisms, etc., which is an area that the center is really focused on from technical. Now, over time, during development or during disease progression, some of these changes will disappear, others will become permanent, primarily in the nucleus, in the form of transcriptional programs, epigenetic regulation, as you hear from Mechanobiology, in terms of three-dimensional chromatin response to mechanical force. So that's what you will hear from Mechanobiology on IRT2 on somewhat intermediate time scales, maybe hours, maybe days. And some of these programs then will be fed back to the cytoskeleton and the extracellular environment, which can cause remodeling over much longer time scales. And in IRT3, the focus will be on exploiting these spatiotemporal transcriptional profiles at the level of the tissue to engineer emerging properties. Typically, you will hear several examples of organoid and tissue on a chip system, both plant and animal from Prina. So that's a summary of what you will hear from us over the next couple of days. Okay, so we've been really productive. You can look at our website. So we have 415 papers to date and a number of reviews, perspectives, and protocols. We are very proud of our educational programs. We have programs for all levels, starting from high school students here, for children and families through our collaboration with the Franklin Institute. For high school students, you'll hear about this later today. For undergraduates in the form of textbooks and courses, and we are developing traveling mechanobiology workshop that many of our four-year college partners and MSIs have been using. All right, so you'll hear a lot more later. And diversity at all levels is a priority of the center. And I say all levels, going all the way from faculty to undergraduates. And we have a number of programs in this regard. I'll just highlight the summer research program for undergraduates and a master's research fellowship where we take students from MSIs and we fund their master's program. And we typically fund students who want to, as well as other programs. So I don't want to take too much time. So if you have any questions, let any of us know. In particular, Annie, our managing director, Avery, who's the director of education and diversity, you'll hear from her later today, and Gwen. So again, welcome again. Weather is not that great. Matter of fact, I got stuck in traffic for like more than an hour. But yeah, hopefully this free time evening you'll get to see the city a little bit, and a lot going on in the city, as well as enjoy the campus.